Assalamu alaikum everybody. I am Mohammad Saleh. My student ID is 2321517. The topic of our presentation is development of potato scar markers to the PVY resistance gene based on a common feature of plant disease resistance gene. At first, we thank to our teacher Mr. Tanim Ahmed. He is the lecturer at Department of Computer Science and Engineering. Let's meet my co-presenter Rifa Sanjida Remo. Our student ID is 2321513. Now we start our presentation. At first, we know that what is PVY, potato virus Y. It is a major threat of potato crops and related solanans species breeding resistant cultivars using wild relatives with resistant genes, such as RY genes, is crucial. However, the lack of essential genes makers for trait selection hampers progress, leading to costly and time-consuming conventional breeding. Recent research has identified scar markers based on ADG2 fragment difference, offering a promising alternative to traditional makers like RFLP, RAPD, and CAPC. Now we see some picture of affected potato. We see the picture A, B, C. Every type of potatoes are affected by PVY. That's our sample. Now, when we process this, identify this PVY, we need to some materials. We need five type of materials, such as plant material, preparation of template DNA, prime design for scar makers, PCR conditions, and at last, analyzing of PCR products. Now, describe of materials. Plant materials. The study utilized a diverse set of plant materials, including common tetraploid potato cultivars, tetraploid and diploid potato breeding lines, and various genotypes with different genetic backgrounds. This include control lines with extreme PBY resistance. In total, 103 genotypes were tested, with 24 exhibiting exceptionally high resistance to PBY. Secondly, preparation of template DNA. Total genomic DNA was extracted from pathogen-free plant grown in vitro using CTAP. Extracted DNA was treated with RNase at 37 degrees Celsius overnight. DNA samples were diluted to 25 TE buffer and stored at 4 degrees Celsius. Then primer design of scar markers. Based on 11 nucleotide difference between ADG2 sequence from primer pairs were designed to especially target ADG2 from with 16 fiber combination a PCR simulating program. Then PCR condition. We saw a chart of a PCR condition. We have primer, sequence, position, length, etc. etc. PCR was conducted in a 50 reaction mixture containing PCR buffer, DNTPs, MgCl2 primer, tag, polymers, and template DNA. The amplification program consists of an initial denaturation at 93 degrees Celsius for 9 minutes, followed by 35 cycles of denaturation at 94 degrees Celsius for 45 seconds. Now, I requested to my co-presenter to come here and complete this presentation. Thank you.
Thank you MD Saleh for giving me the opportunity. Hello everyone, this is Rifa Sanjita Remo ID 2321513. Now I am continuing the rest of the presentation. Now we are talking about analysis of PCR products. Existence of amplified products was checked electrophoretically on a 2% agarose gel used 1x TAE buffer. After electrophoresis, gels were stained in ethymedium bromide distained with distilled water, visualized with immergimustard VDS, and photographed using thermal imaging system for data analysis. Reproducibility of PCR experiments was confirmed at least twice. Now, results. Identification of primer combination for scar marks. Two primer sets produced specific PCR products with no amplification in another genotypes using a scar primer set. Now we are talking about applicability test of scar markers. RYSC3 accurately identify RYADG gene presents in 14 genotypes with reduced DNA concentration needed for one. RYSC4 detected RYADG but also occurred in non-extreme PVY resistant genotype, showing a 96.1% correlation. These scar markers were specific to RYADG from S. tubersome sub-sp and D. jna. Discussion. Advancement in plant molecular genetics have led to development of PCR-based molecular markers for crop breeding, including RAPD, SCR, CAPS, and AFLP. Among these, SCR markers have proven highly valuable due to their simplicity and speciality. In this study, SCR markers, particularly RYSC3 and RYSC4, demonstrated especially accuracy in detecting in RYADG gene in diverse potato genotypes, making them powerful tools for breeding programs. These markers also help identify resistance to other pathogens linked in RYDG. Furthermore, efforts to clone the RYADG gene are underway, potentially shedding light on its molecular characteristic and polygenetic relationship in Solanus. Conclusion. The study expressed gratitude for the provision of DNA samples from multiple sources of acknowledgement, the valuable contributions and support from various individuals and funding agencies in conducting this research. Thank you for your patience, and if you have any question related to this topic, feel free to ask us.